Hello, and welcome to CMA Your DNA, a bioinformatic approach to discovering new ancestors. This is the first of two parts, Part 1 being an introduction to correlated multiphasic analysis. Part 2 will follow in a separate video available on the Discovering New Ancestors YouTube channel or via our website at discoveringnewancestors.com. For those of you unfamiliar with the term, bioinformatics is a subdiscipline of mathematics, biology, and computer science concerned with the acquisition, storage, analysis, and dissemination of biological data, most often DNA and amino acid sequences. In a laboratory, scientists might employ bioinformatics to predict how proteins fold themselves together in 3 dimensions, or to analyze the behavior of newly engineered compounds. However, within our specific context, we'll apply operations and principles of Axiomatic Set Theory to commonplace concepts from genetic genealogy, and in the process, explore a few ideas of our own. Before we proceed further down this rabbit hole, a few legal particulars. The CMA method is a patent-pending process of discovering new ancestors. All rights are reserved, and this presentation remains the intellectual property of discovering new ancestors. Permission is granted for personal, non-commercial use of the CMA process via desktop spreadsheet tabulation using the CMA Master Workbook, which is presented in the second part of this video. Commercial and database management system implementations of CMA require prior written approval from Discovering New Ancestors. Hand-drawn illustrations by Charles Vincent are used with permission. So, just what is correlated multiphasic analysis? Well, we're glad you asked. Correlated Multiphasic Analysis, or CMA as we call it, grew out of a desire to formalize a number of basic principles that genealogists intuitively grasp yet fail to fully exploit in the course of their investigations. Strictly speaking, CMA applies Axiomatic Set Theory to model genetic inheritance in order to facilitate the computational analysis of in-common-with matches. Over the course of development, one particular analogy regarding CMA's mechanisms and methodology became particularly useful in getting the idea of CMA across without getting mired in minutiae. Let's begin with a little background. Genealogy is replete with brick walls. There's a term so common it requires little explanation. But whether it's because of common surnames, missing, damaged, or hard-to-find offline records, untraceable immigrant ancestors, adoptions and non-paternity events, or even the 1870 brick wall, which impacts the ancestry of so many Americans, everyone's family tree has them. Over the years, we've tried various ways of breaking through those brick walls, but every pedigree eventually comes to an ancestor where things just stop. And in many cases, those brick walls are frustratingly close. But whatever the particulars might be, intuitively, it makes sense that a solution to each nagging query must be out there, somehow, somewhere. Autosomal DNA testing is like opening up a window on that wall. And indeed, the very idea of consumer DNA testing, developed within our lifetime, can seem nothing short of miraculous, and remains a source of powerful and transformative discoveries for many individuals and their families. But how do we make sense of what we find? Common ancestors and shared DNA segments are like a roadmap to the past, and in many instances, shared linkage, in conjunction with well-researched family trees, is all that is required to discover new family lines and ancestors. Where circumstances permit, probabilistic models can effectively eliminate unlikely relationships and guide our investigations towards the most plausible outcomes. And when those methods fail, granular strategies, like chromosome browsing, phasing, mitochondrial and Y-DNA testing, and even whole genome sequencing, allow you to zoom in and promise to bring you closer to your origins than you may have previously thought possible. But when you want to study something far away, like a distant ancestor, you don't want a microscope to zoom in. You need a telescope. And so I built one. Not a physical apparatus, but a bioinformatic process for visualizing and organizing collections of autosomal DNA matches across the entirety of an individual's ancestry. It's called CMA, short for Correlated Multiphasic Analysis, and it's provided me with tools for knocking down 20-year brick walls, inquiries which have been resistant to investigation through traditional methods. In the case study we'll undertake in the course of this video, an 11-point analysis via CMA produced strong evidence of a Jewish ancestor amongst my great-great-grandmother's pedigree, in addition to identifying hundreds of DNA matches connected via her ancestral lines. To develop our analogy, just like a telescope, CMA collects and correlates data over a large region. CMA only shows you what's already there, but clearer. CMA doesn't require specialized scientific knowledge to use, and CMA does one thing, but does it really well. Think of how, when you look at a star, its light travels a long, long way and shines over a large part of the world, even though our eyes are small and only take in a little light. A telescope works just like your eyes, but gathers more light from a larger lens. Now, think of a distant ancestor and how the DNA you've inherited from that ancestor is like the star's light, transmitted across generations rather than across physical distance. Likely as not, you've received only a small amount of any distant ancestor's DNA, but some of that genetic material is potentially available to every living descendant of your ancestor, the way a star's light can be seen over a large area. If only there were a way to collect information about genetic inheritance from a larger set of descendants than just a single person, and to make sense of that information once we have it. Well, there is a way, and that's what CMA does. The CMA process correlates the entirety of your DNA matches against the matches of other family members to provide otherwise unobtainable insights into your ancestral lines. CMA's advantages. CMA delivers powerful insights drawn from the totality of a subject's DNA results rather than just the top 1 to 5% of matches. CMA leverages large collections of autosomal DNA matches with or without associated family trees. CMA correlates member matches beyond the reliable 5 to 6 generation or 200-year window otherwise available through segmental analysis of DNA. CMA does not require any additional processing of raw DNA data. CMA does not assume any advanced scientific knowledge on the part of the end user regarding chromosomes, genetics, alleles, or the like. CMA rewards the targeted testing of immediate and extended family members. CMA lends itself to an interactive click-driven interface. CMA is dynamic and multiphasic, reframing its solutions as additional member matches and/or correlating criteria are added, which is to say that every data point and test subject interacts with every other data point and test subject, so that when you add or change something, everything instantaneously adjusts to reflect this new information. CMA quickly identifies non-paternity events without impacting the quality of its core findings, so bad data won't skew your quality data and may even heighten the distinction between useful and irrelevant information. CMA supports intuitively structured investigations and queries, so you can start with what you know and work towards your brick walls. CMA is accessible to anyone with an appreciation of the concept of ancestral family lines and common ancestors. CMA provides raw materials for follow-up investigations via traditional methods. CMA can specifically address the genealogical brick wall challenges faced by individuals with non-paternity events, unknown ancestors, or unknown parentage, with immigrant ancestors, and for marginalized populations whose ancestors might not appear by name on U.S. censuses prior to 1870. CMA can also impute a genealogical relationship by comparing the patterns, correlations, and correspondences of an unknown subject's autosomal DNA matches with those of established genealogical relations. Finally, CMA is axiomatic. What this means is that CMA uses Axiomatic Set Theory to model genetic inheritance as a collection of intersecting, shared, and complementary sets. Axioms are descriptors and rules which define the boundaries and operations of a mathematical system. These axioms allow CMA to be modeled via a desktop spreadsheet environment or to be incorporated into the back end of a database management system. Axiomatic systems can also form the basis for machine learning and artificial intelligence, so that the basic analytical support furnished by the spreadsheet and DBMS implementations of CMA are potentially only the beginning. We'll demonstrate an axiomatic model of direct descendants at the end of Part 2 of this presentation. Want to know more? Let's begin by considering two individuals, A and B. A and B are first cousins, a relationship verified by their shared DNA. So in addition to around 900 centimorgans of linkage, A and B also share a set of grandparents, their most recent common ancestors, or MRCAs. Beyond a set of grandparents, A and B share each and every ancestor of those grandparents. Next, let's consider three other individuals, C, D, and E, each of whom share DNA with both A and B. How might C, D, and E be related to A and B? There are actually two ways an individual could share DNA with both A and B. Either the individual is descended from one or both members of A and B's MRCA couple, which is the case of hypothetical C, or the individual shares a common ancestor with a member of that MRCA couple, which is the case of individuals D and E. Take a moment and pause this presentation if you like to review each of these points, as they're fundamental building blocks of CMA, two of the basic principles that genealogists intuitively grasp. We'll revisit this scenario at the end of Part 2 to explore a third possible connection to A and B and demonstrate how the axiomatic method can differentiate among these connections. When individuals share the same literal segment of DNA, we speak of them belonging to a particular genetic network, which is a reasonably specific collection of DNA matches. Ancestry's shared matches are an example of this. Let's instead consider the set of all DNA matches which A and B share, otherwise known as in common with or ICW matches. We'll call this set of shared matches the genetic complex of A and B, and write that as complex AB, or the intersection of A and B. Let's return to our diagram and shade in the complex about A and B. As a reminder, all individuals in the shaded area are either descended from one or both of A and B's shared grandparents, or share a common ancestor with one of their grandparents. Further, note that the type of connections made by D and E could occur to or from any ancestor of either most recent common ancestor. Finally, since every DNA match shared by A and B is related through their most recent common ancestors, we can label the complex about A and B as the complex about the most recent common ancestors of A and B. When we considered individuals C, D, and E as genetic relations to A and B, we assumed them to be hypothetical, but let's instead allow that they are individuals with known relationships to A and B, and see what, if anything, is changed by those genealogical connections. If we consider C to be A's niece or nephew, and B's first cousin once removed, very little changes with regards to the complex about the most recent common ancestors of A and B, as A, B, and C share the same most recent common ancestors. This is not to imply, however, that nothing changes. Because A and B share most recent common ancestors with B and C, the genetic complexes about A and B and C are identical. When we consider the genetic complex about A and B, the addition of individual C adds DNA matches to our complex, as we now consider the matches B shares with C and the matches shared by all three subjects, in addition to our original A and B matches. What's more, the addition of individual C further supports rethinking the complex about A and B as a complex organized about their most recent common ancestors, since only one intersection set in our Venn diagram consists of A and B exclusively. Note that A and C share a most recent common ancestor of their own, We'll revisit this later, but it's worthwhile to consider that some of the DNA matches shared by A and C exclusively may originate among the ancestral lines shared with B, while many of the matches shared by A and C have their origins among ancestral lines not shared by B. Now, consider D, in this instance, a fourth cousin to A and B. The genetic complex around the most recent common ancestors of A and D is a proper subset of the complex around the most recent common ancestors of A and B, and does not include individual E, since individual E originates along a different ancestral line. Because individuals A, B, and C all share the same most recent common ancestor with subject D, the complexes about any of A, B, and C with D are functionally equivalent. This suggests that DNA matches common to A and D, B and D, C and D, and combinations thereof, ABD, BCD, ACD, and ABCD, so long as they include individual D, are all members of the complex about the most recent common ancestors of A and D. These letter name combinations are called classifications, and we'll explore their significance more fully in the course of this presentation. Finally, the complex about the most recent common ancestors of A and E, a second cousin once removed to A and B, is also a proper subset of the complex about the most recent common ancestors of A and B. But, when compared to the complex about the most recent common ancestors of A and D, is less focused, including more ancestral family lines. This suggests that the closer the relationship among constituent matches, D being a fourth cousin, whilst E is a second cousin, the more diffuse, or if you will, less focused, the collection of DNA matches becomes. We'll revisit this momentarily. Our investigation thus far has provided us with several takeaways, what in set theory we'd call axioms. Axiom 1, individuals who share DNA also share a common ancestor. In this case, A and B share around 900 centimorgans of linkage and are cousins. Therefore, they share a set of grandparents. Axiom 2, when a common ancestor for two or more individuals is known, a genetic complex about that ancestor or couple may be defined to include all DNA matches common to any combination of the descendants of that ancestor or couple. In our diagram, this means that all member matches shared by any combination of individuals A, B, and C will be members of the genetic complex organized around their common ancestors, and that member matches D and E will only be included in this complex if they match two or more of A, B, and/or C. We'll carve out a method for managing the shared matches of closely related individuals, such as A and C, in the second part of this presentation. Axiom 3. Genetic complexes about shared most recent common ancestors are identical. Therefore, the complex about the most recent common ancestor of A and B is identical to the complex about the most recent common ancestor of B and C, which is also identical to the complex about the most recent common ancestor of A, B, and C. Each of these complexes is distinct from the complex about the most recent common ancestor of A and C. Axiom 4. A genetic complex, which includes several individuals sharing the same most recent common ancestor with an outlier, will include all non-trivial match combinations of those individuals with the outlier. Therefore, the complex about the most recent common ancestor of A and E is identical to the complex about the most recent common ancestor of B and E, which is identical to the complex about the most recent common ancestors of B, E, and C, which is identical to the complex about the most recent common ancestors of A, C, and E. Strictly speaking, the order of letter-named individuals does not matter. The complex about the MRCA of BEC is identical to the complex about the MRCA of CBE, but for consistency, we'll prefer an alphabetized arrangement wherever possible. Axiom 5. If a genetic complex embeds multiple individuals with shared most recent common ancestors, the way the complex about the most recent common ancestor of A and D does with respect to A, B, and C, then that complex will be a proper subset of the complex defined by the embedded elements only. Therefore, if A, B, and C share a most recent common ancestor, and D shares an ancestor with that MRCA, then the complex about the MRCA of A and D will be a proper subset of the complex about the MRCA of A, B, and C. The basic idea here is that the genetic complexes of more distant ancestral lines are contained within or included in the complexes of more closely related most recent common ancestors, so that when we correlate the matches of third or fourth cousins, and matches which were previously assigned to a closely related complex now appear in a more distant collection, we're not getting a different solution so much as a more accurate or focused solution. Axiom 6. Genetic complexes defined by distantly related individuals will be more focused or less diffuse than those of close relations. Therefore, the complex about the most recent common ancestor of A and D is more focused than the complex about the most recent common ancestor of A and E, which in turn is more focused than the complex about the most recent common ancestor of A and B. This is basically a reiteration of our previous axiom regarding the inclusion of collections, but also speaks to the comparison of collections from different ancestral lines. Axiom 7. Genetic complexes defined by individuals from separate family lines about a common descendant will be disjunct subsets of the complex sharing that intersection and their descendants. Therefore, the complex about the most recent common ancestors of A and E, and the complex about the most recent common ancestors of A and D don't overlap each other in the complex about the most recent common ancestors of A and B, exclusive of the descendants of that most recent common ancestor couple. Bear in mind that the whole point of these axioms, or rules, is to describe the phenomena of autosomal DNA matches in the language of mathematics, in our case, set theory, so that we may model and analyze that phenomena, or system, if you will, in consistent terms, which may ultimately allow a computer program to assist us in our queries. In part 2 of this presentation, I'll demonstrate a scripted environment in Microsoft Excel constructed specifically for these purposes. Admittedly, the preceding points are a lot of theory to absorb in so short a span, so we'll add just one more construct before attempting a practical study. Let's call the set of matches shared in common by any of the individuals participating in our analysis the Analytic Control Set, or ACS for short. We'll use the Greek letter theta to represent our analytic control set in diagrams and formulas. For two test subjects, A and B, the analytic control set is equal to the intersection of A and B. For subjects A, B, and C, the analytic control set equals the intersection of A and B, the intersection of A and C, the intersection of B and C, and the intersection of A, B, and C. As we increase the number of test subjects under consideration, we can state that for any collection of test subjects, the analytic control set remains the set of DNA matches shared by two or more subjects within that collection. Lastly, the number of elements in any collection is known as its cardinality and is notated with solid bars on either side of the collection's name. But enough with all this theory. Let's apply what we've learned in practice. Consider DS, a member of my extended family. DS took an autosomal DNA test with a well-known provider and received results of just over 5,100 shared matches of 10 centimorgans or greater. We'll represent him as such. DS will be the nexus of our investigation, our target individual, and the subject A of our case study. I'm connected to DS through his father's side, so I already have a reliable paternal pedigree for him. The brick walls in this pedigree represent figurative impasses for which, even after a dozen or more years, little or no reliable information, documentation, nor DNA matches have been found. In the case of wall A, even Y-DNA tests have failed to yield actionable insight as to how this family's Shaw line might be connected to the ancient Scottish clan or to any other pedigree. Our case study will primarily investigate wall C, the unknown ancestors of DS' great-grandmother Catherine Mardell. You'll recall from our preliminary discussion that genetic complexes are organized around most recent common ancestors, and that we can derive some sense of the shape and density of these complexes based on how many generations removed the organizing most recent common ancestor of each complex is from our nexus. For this reason, let's examine DS pedigree and assemble a table of complexes. From the diagram, you can see DS and his spouse at generation 0, DS parents, 1 generation removed from DS, DS paternal grandparents, 2 generations removed, the Shaw-Howell couple, 2 sets of great-grandparents, 3 generations removed, the Shaw-Mardell couple and the Howell-Butler couple. 4 generations removed, we have the Shaw-Jones couple, and since we don't have any ancestors for Catherine Mardell, we can indicate her family group with a 4th generation Mardell. At the top of the pedigree, we see the Shaw-Mackintosh couple. We'll need the results of DS autosomal DNA test, all 5,100 matches. The table at right has been filtered to display matches from the upper rank of DS paternal line, corresponding to the pedigree shown previously. The matches shown with bold identifiers will be used in our case study as we have obtained their DNA matches for comparison with DS. Access to the full roster of matches in common with DS DNA results would facilitate a more streamlined and thoroughgoing CMA experience, and such access would be possible if CMA were incorporated into the back-end reporting of DS Test Provider. Generally speaking, CMA utilizes member matches of 1,800 centimorgans and less. The member match in the topmost rank is CG, DS' first cousin, who shares 766 centimorgans with DS. Let's designate CG as our subject B and begin our analysis by comparing CG's 6,700 DNA matches with DS. As first cousins, DS and CG share a set of grandparents. We'll use the surnames of their grandparents to label their shared genetic complex as complex Shaw-Howell. Our initial test subjects provide us with an analytic control set of 379 matches, which include, in addition to DS and CG, 377 other members sharing their common ancestral lines. Recall from our preliminary investigation concerning the hypothetical connections of any 2 individuals that the matches common to DS and CG must either be descendants of their Shaw-Howell grandparents or must share a common ancestor with either grandparent. If we average and rank the shared linkage of the member matches common to DS and CG, we find our next test subject, GK, a half-cousin with almost 24,000 matches of her own. As a half-cousin, GK shares but a single grandparent with DS and CG, and so any DNA matches GK shares with DS or CG must be through the descendants or ancestors of Florence Howell, whose maternal ancestors were the Butler family, forming the complex Howell-Butler. As such, complex Howell-Butler is composed of the matches common to subjects A intersecting C, B intersecting C, and the match is shared by all 3 test subjects, which yields a total of 686 member matches. 134 of these matches were previously assigned to complex Shaw-Howell, which is now reduced from 379 elements to 245. Correlating GK's matches expands the analytic control set from 379 elements to 931, and partitions what was exclusively a collection of 379 Shaw-Howell matches into a set of 245 complex Shaw-Howell members, complemented by 686 complex Howell-Butler members. Our next test subject from the roster of DS paternal DNA matches is BH, a second cousin once removed on the Shaw line. The addition of BH brings us closer to our brick wall. At this point, things begin to get interesting. From the Venn diagram, we can see that CG has inherited more DNA from the Howell line than her cousin DS, whose matches are heavily weighted towards the Shaws. Let's review our complexes. Since BH, individual D, is our most distant connection to complex Shaw-Mardell, any match D shares with the other test subjects must be assigned to complex Shaw-Mardell, and so the population of complex Shaw-Mardell is 762 plus 17 plus 28 plus 5 plus 6 plus 17 plus 79, which equals 914 matches. Likewise, GK's shared matches definitively connect those individuals to the Howell lines, so the population of complex Howell-Butler is 658 matches. Finally, the 228 matches shared by DS and CG exclusively comprise complex Shaw-Howell, a set of matches which could ultimately resolve as either Shaw-Mardell or Howell-Butler matches. Our analytic control set consists of the union of all three complexes and contains 1,800 matches. To review our hierarchy of matches from the top down, our universal set contains every DNA match of every test subject. Thus far, our universal set contains 44,635 matches. The universal set is composed of the matches of individual test subjects. Subject A is designated as the nexus of the investigation. Additional test subjects are typically selected at the outset of our analysis from Subject A's roster of matches in accordance with the following criteria: subjects with known genealogical connections to our nexus, subjects with greater average shared linkage, subjects with connections to other members of the complex under investigation, and lastly, subjects with well-researched pedigrees. Theta is our analytic control set and is composed of all duplicated or shared matches among our test subjects, 1,800 matches so far. The elements of the analytic control set are the only member matches which directly participate in our CMA process. The analytic control set is subdivided into genetic complexes, which are defined by the most recent common ancestors of the most distantly related test subject participating in a shared match. Each complex is notated by a complex symbol and the surnames of the most recent common ancestors shared by its participants. Examples of complexes in our study are complex Shaw-Howell, complex Howell-Butler, and complex Shaw-Mardell, but the composition and arrangement of genetic complexes will vary from investigation to investigation. Genetic complexes are composed of matches grouped into ACS classifications. Classifications allow us to categorize matches using letter names. For instance, complex Shaw-Mardell consists of matches which include subject D, 762 AD matches, 28 BD, 17 ABD, and 5 ABCD matches, amongst others. Each classification consists of DNA member matches which are shared among our test subjects in consistent ways. For example, matches shared by subjects A and B only, or matches shared by B and D only. While ACS classifications may share a most recent common ancestor, and therefore belong to the same genetic complex, the internal similarities of each class suggest that matches within a given classification represent shared DNA from a particular ancestral line or set of lines. This is reasonably profound in that individual ACS classifications allow us to effectively pull apart or stratify the large collection of member matches which compose a genetic complex. Since the matches in any genetic complex typically represent members who share a common ancestor with the most recent common ancestor couple, each analytic control set classification represents a facet or subcollection of the ancestors of that most recent common ancestor or couple. Regional or ethnic properties of the matches within a given classification may provide us with further insights to support investigations by other means, and correlating the matches of an isolated ACS classification with additional test subjects taken from within that classification may in turn further isolate ancestral lines, as we will explore in Part 2. Individual DNA matches are the granular elements of CMA, which are correlated and tallied among test subjects of known and unknown genealogical relations. The CMA process assigns an ACS classification and corresponding genetic complex to every DNA match participating in the analytic control set. Moving down the roster of DS paternal DNA matches, we encounter JM, her niece DE, and an assortment of their immediate relations who share DNA with our Shaw family subjects. The classifications of the matches JM and DE share with subjects A and B suggested a connection along the lines of subject D, but a more distant relation. A case study, available at the Discovering New Ancestors website, reveals JM and DE's Shaw connection as descendants of a daughter fathered by DS' great-grandfather prior to his marriage, making John Shaw, 1811 to 1890, the sole most recent common ancestor of the genetic complex defined by their matches. Knowing the names of John Shaw's parents, we can label this complex as complex Shaw-Jones rather than simply complex Shaw. A Venn diagram no longer suffices to tabulate the intersections of 6 test subjects, but we'll review an alternative tabulation methodology in the second part of this presentation. Further down the roster of DS matches, a second collection of DNA matches, which includes family relations of member MB, appear to be descended from the offspring of John Shaw and another woman prior to his 1864 marriage. A separate case study, available at the Discovering New Ancestors website, sets forth the particulars of MB and family's connection to the Shaw line and complex Shaw-Jones. An additional test subject, TC, does not have direct knowledge of her paternal pedigree. However, the pattern of member matches she shares with subjects A through G strongly suggest that John Shaw is also a direct ancestor on her paternal lines as well, most probably a great or great-great-grandfather. In order to better determine the precise nature of her connection to the Shaw line using CMA, TC would take the set of matches she shares with our test subjects and use them as a preliminary analytic control set, selecting additional test subjects for correlation from that same ACS. Correlated together, subjects E through H help delineate the set of member matches associated with brick wall C by removing some matches previously assigned to the Shaw-Mardell complex and associating them instead with the Shaw-Jones complex. 3 additional test subjects round out our survey of DS paternal relations. Subject I is descended from John Shaw's brother and completes our complement of complex Shaw-Jones matches for the time being. Subjects J and K are descended from John Shaw's uncle and support the assignment of matches to complex Shaw-Mackintosh. Before moving on, let's review the composition of our genetic complexes. Complex Shaw-Howell is the set of DNA matches shared by A and B only. Although our preliminary analysis has assigned 180 member matches to this genetic complex, it's worth remembering that complex Shaw-Howell includes both direct descendants of DS paternal grandparents as well as members who share a common ancestor with either grandparent. As such, the great majority of the 180 elements of complex Shaw-Howell will ultimately be assigned to another of the Shaw complexes or to a Howell family complex if they can be shown to match a descendant of that line. Complex Howell-Butler is the set of DNA matches shared by A and or B with subject C. Complex Howell-Butler is formed from the intersection of complex Shaw-Howell with the DNA matches of half-cousin GK. Because GK shares just a single most recent common ancestor with the members of complex Shaw-Howell, any DNA matches shared by A or B with C must be members of complex Howell-Butler. Correlating additional descendants of our Howell-Butler couple against complex Shaw-Howell would be a useful way to fully stratify this complex. Complex Shaw-Mardell is the set of DNA matches shared by A and or B and subject D. Correlating the matches of subject D removes matches from complex Shaw-Howell and creates a complex whose elements are connected through either more distant Shaw relations or through our brick wall C. Correlating additional Shaw matches will assist in making a final determination as to which complex each match is ultimately assigned. Complex Shaw-Jones is the set of DNA matches shared by any of E, F, G, H, and or I, and any other elements of A, B, D, E, F, G, H, and or I. As with correlating subject D, this tier of subjects removes matches from complex Shaw-Mardell and creates a complex whose elements are connected through either more distant Shaw relations or through our brick wall B. Complex Shaw-Mackintosh is the set of DNA matches shared by J and or K and any subjects other than C. And once again, elements of existing Shaw complexes migrate away from our nexus to further stratify our analytic control set. Thus far, we have tabulated over 132,000 DNA matches from 11 test subjects to form an analytic control set of around 8,000 elements in pursuit of actionable information about any of DS' ongoing brick walls. However, despite the number of uncorrelated subjects left in DS roster, this exercise amounts to little more than a proof of concept without some kind of analytical takeaway. Let's see what we can find. Ancestral brick wall C concerns the pedigree of DS' great-grandmother Catherine Mardell. Our analysis includes 3 descendants of Catherine, subjects A, B, and D. Following the tabulation of subjects A, B, and C, the addition of subject D to our analysis enlarges our analytic control set to around 1,800 matches, roughly 1,000 of which are members of complex Shaw-Mardell. However, this 4-subject analysis lacks any means through which we might determine which of these 1,000 matches are ancestral Shaws and which belong to the Mardell line. Our solution lies in the selection of our subsequent test subjects, E through K. Because each of those subjects is connected to the Shaw line, the effect of these additions is to enlarge the analytic control set both in size, from roughly 1,800 elements to 8,000, and also in variety, populating more distant complexes and reducing the size of complex Shaw-Mardell from roughly 1,000 matches to a number closer to 850. The diminution of complex Shaw-Mardell downwards to 850 matches implicitly brings us closer to a complex Mardell solution set because the cumulative effect of the thousands of matches from Shaw-related test subjects E through K is to pull Shaw matches out of complex Shaw-Mardell and into complex Shaw-Jones and complex Shaw-Mackintosh. Similarly, increased representation of test subjects taken from known relations of the Howell family line would have the effect of pulling latent Howell matches out of classification AB, leaving the remaining Shaw matches in that class likely participants in the complex Mardell as well. We'll discuss how best to analyze and develop ancestral lines within complex Mardell in Part 2 of this presentation, but a preliminary survey of the ACS classifications embedded within this genetic complex suggests several possible avenues of investigation. Complex Mardell consists of DNA matches shared by subjects A and B with subject D, comprising ACS classifications AD, BD, and ABD. The AD classification within complex Mardell is conspicuous in that many of the matches within this classification appear to share Ashkenazi, Eastern European Jewish, surnames, an ethnicity which is notably absent among the other classifications in complex Mardell and elsewhere in our analysis. suggesting Catherine Mardell likely had an ancestor of Jewish extraction. Surveying the elements of complex Mardell beyond this classification provides us with a collection of family lines which must, in some way, connect to our Mardells, although determining a definitive connection from the pedigree of any of these matches would be nothing short of a miraculous stroke of luck. Nevertheless, a close reading of the family trees associated with these member matches suggests several family lines which might lend themselves to investigation. Given the lack of any actionable information regarding the Mardell family lines over the course of 20 years' research, CMA's ability to suggest evidence of a heretofore unknown Jewish ancestor, in addition to the hundreds of member matches identified as prospective connections, augurs well for its future prospects. In Part 2, we'll explore correlated multiphasic analysis on the desktop platform via the CMA Master Workbook, a scripted application which employs Microsoft Excel to create a robust environment in which to perform CMA. We'll also attempt a secondary CMA analysis using a genetic complex taken from our Analytic Control Set and establish CMA-oriented axioms regarding the properties of direct descendants. For a link to Part 2 of this presentation, and for further CMA resources, visit us at discoveringnewancestors.com.